Hello. I would like to welcome you all to the next presentation in the Behavior and Cognition track of the Bio Conference Neuroscience meeting today. My name is David Kennedy. I'm at the University of Massachusetts Medical School. I co-direct the Child and Adolescent Neurodevelopment Initiative with Dr. Jean Fraser, who is head of child psychiatry there. And I run our division of neuroinformatics. I'll be speaking today on a number of topics related to big science, big data analysis in behavior and cognition, and particularly look at the neuroimaging and informatics aspects of the resources that are available for such an endeavor. So the first topic to consider together is what is big cognition. Big is one of these popular terms, these days. Big data, is very popular. The NIH has a new disciplinary efforts in big data to knowledge, and big data is, there's new journals in big data, so big data is sweeping, the industry. So when it comes to sort of cognitive science and behavioral science, what types of things are big? Well, to some extent, one of my simplistic visions of what big data is nowadays is anything that's an order of magnitude larger than when I was a graduate student. To me, counts as big data. If when I was a graduate student, a study was done with 20 subjects, and nowadays, it's 200 or 2,000 is the appropriate number of subjects, well, we've moved into the big data era. If an MRI scan when I was in graduate school, was 6 megabytes, and now a MRI session is 200 gigabytes, well, we've moved into the big data era for our particular domain. When I was a graduate student, cellular images, electron microscopy, would be for a cell would be a few hundred megabytes, and now the high resolution full reconstructions of cells in electron microscopy is terabytes of data. So a lot of the domains now all have to face with their own version of big data. In cognition and neuroimaging aspects of cognitions, we are now doing much larger acquisitions per each of our subjects, and many more of these subjects. Our analysis is becoming more automated, and we're deriving more information from each of our subjects, and we need to manage and control all that information. So one of the other aspects of big data is our own data is getting large, but also data sharing is beginning to happen. So not only do we have to worry about our own data, we have to worry about other people's data, and how do we relate our data to their data? How do we pool those data? How do we infer higher order information from the multiple data sources that are at our disposal? So the good news, of course, is data sharing is happening. There is especially in the neuroimaging communities, there's a lot of data sharing that's going on that we'll look into a little bit more further in this discussion. Thanks in a large part to NIH mandates for data sharing and publisher mandates for data sharing, the scientist, perhaps somewhat begrudgingly, is moving into the data sharing era. As the data becomes available, there are many types of data. Thus, there are many different types of sources where that data can be shared. And these sources are getting large, and they're very distributed. Depending what type of data you're looking for, it could be in any of many different locations. The other aspect and rationale for a lot of the big data and big data sharing that's going on is a sort of a backlash against sort of smaller end studies and small numbers of subjects that may be perceived to be underpowered, and often in many domains there's not always complete replication or easy replication of different findings. So again, picking on neuroimaging a bit, and in certain communities, many different papers might say the white matter is big, the white matter is small, the white matter is no different between patient group X and patient group Y. And if each of the studies are small-ish and there's a lot of variability in subject selections, there's just more ambiguity about what the right answer is to many of these topics. So again, to help not prohibit small local studies from happening, but to also begin to be able to recover larger ends and larger powers through the ability to do data sharing and pool data together is important across all the different disciplines. And again, one can think a little bit back to the evolution of genetic analysis. Again, in the old days, it used to be okay to do your large genetic GWAS study, but nowadays you have to do your study and replicate that in a couple other big data sources before you can publish that. And so while the neuroimaging, the cognitive and behavioral communities aren't quite to that point yet, I think that's a good concept to keep in mind, that again, as we do our studies, we need to look to the opportunities to see replications in the other shared data of each of our findings. So as the approach to larger data sharing and larger big data approaches begin to evolve, the tools and resources that we as cognitive neuroscientists or behavioral neuroscientists or neuroscientists in general, there's an evolving set of tools that is changing to meet this big shared integrated landscape. And so again, much of my talk today will be looking at some of those tools and resources that are available to navigate this landscape. So just as a brief overview of what we'll talk about over the next 45 minutes or so, we'll look at how do we find these various resources. Resources is very broad in this context. It can be data, it can be software, it can be other types of things. Once we've found these resources, what do we do with these resources? How do we use them? Often, if they are data resources, we have issues about how to do data analysis with those, and so there's computational topics that raises for us. So often, we traditionally will bring these to our own ground computers, to the computers that are in our own local control or our own local institutions, and that's fine. But again, emerging technologies for cloud-based computational environments is coming along, and so we need to keep abreast of the developments in that, so that we know the right resources for our various computational needs. And then we'll spend a last few minutes of the presentation looking at a couple case studies, a couple examples drawn from more real-world types of examples. So I've already suggested that there are many resources out there, and again, many resources of different types and many resources housed in different environments in different locations. So the neuroscientist, the cognitive neuroscientist, needs techniques to find the resources that are available for whatever particular situation they need to deal with. So let's look at some of the options for finding resources. Well, let's first think about what just the examples of what that breadth of resources are that are available. And so again, in slides, I'm not going to be able to show you every resource known to humankind out there, but I pick a couple examples of resources that they are important and famous, and sort of contain the information, some of the information that one wants to keep track of. So thinking about derived data, and so not worrying about the raw data yet. Often when people do experiments and do their analyses, they end up with results that become the hallmarks of their publications. But the publications and the conclusions from those publications are often drawn upon specific analytic results. So we think about functional MRI, for example. Much of the processing and endeavor in a functional MRI experiment is to derive foci of activation, so locations in the brain where something about the blood flow or the bold signal is changing in response to some sort of changes in the challenge that the subject is being faced with. So over the 2 decades of functional MRI research and thousands and thousands of papers, hundreds of thousands of these spots, of these foci of activation, have been identified and published and put into the public domain via the scientific research enterprise. So back when the literature was small and 10 or 20 papers, a researcher could hold on to that information all in their head and understand what the community was telling them about the functional MRI of a particular topic. But as again, the literature and the number of papers that talk about different activation studies explodes, you need computational ways of managing this derived information. So there's many resources that have been developed to help the investigator manage this information. I list 3 of them or 4 of them here. The Brain Map database, a very famous and one of the oldest of them that again is a database of the foci of activation as published in the literature, so that one can integrate the experiments, conditions, the tasks, the other properties of the experiments, and compare and contrast them amongst the different publications. SumsDB also came along more recently, but again has a very similar challenge to get all the foci of activation or foci of volumetric gray matter density differences and manage these foci in a particular common coordinate system. And then the Breed Wiki and Neurosynth are additional resources that, again, given a location in space, you can report what you've seen there, and then query the literature, so to speak, as to what other people have seen anything there and the types of characteristics that are evocative of those particular locations. So even just within a small concept, one piece of the types of cognitive data portfolio, the foci of activation, there are many different resources that can house this information, and different ways that the investigator needs to have in order to access all this information. And again, foci of activation is just one of many different derived entities that come from neuroimaging data, and then there's other data sources as well. But within our neuroimaging literature, often people will go out and make volumetric calculations of different brain structures. And so again, thousands of publications talk about the brain volumes of this, that, and the other structure in this patient population and that patient population. So again, we need techniques to house that derived data sources, so that one can not have to remember all that and be able to integrate and compare across those different publications. And so the Internet Brain Volume Database, IBVD, is one of those types of resources. Neuroanatomy and just the textual referencing of neuroanatomic in relationship, neuroanatomic concepts in relationships to different publications is fairly frequent. So Russ Poldrak and the Austin, Texas group have created PubBrain, which generates relationships between publications and brain anatomy. Connectivity is very important. Just saw a nice presentation on the Human Connectome project a few hours ago. But again, lots of connectivity measurements are being made in humans and in the macaque, and we need ways to aggregate that information across the different publications that come out. COCOMAC is a classic connectivity database for the connections as observed in the macaque, and then Connectome Wiki and other newer connectome databases are coming along to aggregate information across those applications. Then in addition to derived data itself, there are derived concepts. So again, thinking about the Cognitive Atlas, another resource that helps tie together the higher order concepts that have been developed. So concepts like, what do different brain regions do? What types of behavioral tasks are what types of domains do they tax, and how do we have set up relationships between multiple different types of working memory tasks, etc. So we need to be able to integrate these higher level concepts as well as the lower level concepts of extracted metadata from the imaging data itself. Speaking of imaging data, that also needs to be shared and made available, and lots of data is currently available. I divide, again, this is just the tip of the iceberg of many, many different resources that help investigators find and share data. I divide these sort of into two different classes of sharing initiatives, one of which the top three listed here are sort of required sharing initiatives, and sort of required in that to participate in the Alzheimer's Disease Neuroimaging Initiative, part of your mandate to participate in that was to share the data within that consortium, and a good bit of funding in those initiatives were made available to make the sharing happen for that particular group. And I just picked sort of three random cases of many other ones that could be available. PING, the Pediatric Imaging, Neurocognition and Genetics resource, 1,400 typically developing kids, again, was mandated to share and was given them money to help make that sharing happen. So in contrast to that, I also like to point out some of the grassroots efforts, people who are sharing data not because they had to or part of their contract to do that, but because they want to and because it's sort of the right thing to do in many ways. And a lot of this comes from the 1,000 Functional Connectomes consortia investigators spearheaded by Mike Millam and Javier Castellanos at NYU and Nathan Klein Institute. Not only so they were getting spearheaded and really took an aggressive leadership role in proactively making resting state functional connectome genomic data available for the world. And it turns out that so 1,400 subjects were shared from 20 some different institutions, and really laid the ground, and was a typical young children through old age, and again really made a great resource of resting state data available to the community. And a lot of effort could go into exactly how do you share and pool data of that type. And it turns out the resting state data is fairly well integrable with the appropriate analytic techniques, that you can actually succeed at getting larger power by the sharing of that resting state data in typical subjects. And that was followed up with similar consortiums spearheaded by Michael and his teams, following up in resting state with ADHD children, the ADHD 200 project, and then more recently the Autism Brain Imaging Data Exchange, or ABIDE, for autism resting state data. And again, these are not people who are sharing because they had to. These are people who are sharing because they want to, and use their different available resources to make that available. And again, I hate to put any up here in some sense, because there are many, many, many others. It's just sort of ones that come to mind. So while lots of data is being made available, there are, for the investigator who does want to share data, there is a number of sort of resources for participating in public data sharing that one can access. I'll speak more at greater length. The NITRC, the Neuroimaging Informatics Tools and Resources Clearinghouse image repository, is one of these public resources. The 1,000 functional connectomes, the ADHD 200 ABIDE, PING, many of the publicly available data sets are made available through that resource. So if one wants to share data and wants to contact NITRC, they can help you work on data sharing plan for that data. NITRC is built off a core infrastructure that's called XNAT, Extensible Neuroanatomic Archive Toolkit, spearheaded by Dan Marcus and Washington University in St. Louis. And they run XNAT Central, which is another place where OASIS, the open access structural imaging and aging lives. One can utilize the XNAT Central resource to share data. Coins from New Mexico, the MIND Research Network in New Mexico makes data available. It also has the ABIDE data and other data sets as well. You contact them, and they'll help you do that. The LANI, the Laboratory of Neuroimaging, now at University of Southern California, their image data archive hosts some very large and major data sets. Such as the ADNI, the Alzheimer's Disease Neuroimaging Initiative, and others. And so again, that's a resource that's available. People contact that provider, and they'll work out a way to share your data. There's other more specifically domain specific silos that are occurring as well. And so one that we'll talk more about later is NDAR, the National Database for Autism Research, which hosts a lot of imaging data, plus all the other behavioral and cognitive and genetic and all the information that's being collected in NIH-funded studies of autism are being mandated to share their information through the NDAR database. And they also host the NIH Pediatric database as well, Neuroimaging database as well. And so again, any effort that's going on in autism, calling up Dan Hall and his team, and they're more than happy to help you figure out how to share your information through that resource. And again, as with all these lists, these are just the tip of the iceberg of many resources that are available. So again, for the person who wants to share their raw data, the person who wants to share their derived data, the pile of resources that are available is becoming quite rich. And again, the investigator then has to figure out how to deal with and comply with these different data sharing types of opportunities. It's also then beholden upon the data sharing providers to try to meet the investigators halfway and make that easy enough and not too burdensome to do, to take advantage of the goodwills and wishes to share the data as well. So again, the blessing and the curse with lots of resources being shared. There is then the problem, more and more problems, from the again end user's point of view of how do you find the information that is relevant. So the proliferation of information sources means we need some sort of aggregation environment, some way to pull information from multiple sources, so that you, the investigator, don't necessarily have to know all yourself what all the imaging available resources are. If your aggregator knows that, then you go and ask your query, and your aggregator can pull information from multiple resources in order to deliver that answer to you. The extensibility of you doing that one resource at a time is just not going to work. So we need these aggregators. We'll talk about two aggregators for a little bit now, first of which is the Neuroscience Information Framework, or NIF, run by Marianne Martone and colleagues at University of San Diego, and also one I'm closely involved with, the Neuroimaging Informatics Tools and Resources Clearinghouse, or NITRC. Both of these initiatives are NIH Blueprint funded initiatives, and have been around for 8 or 9 years in these cases, and again have slightly different mandates to provide different types of information and aggregation to the community. So jumping right into NIF, the mandate of NIF is really quite broad. Every resource sort of known to humankind that is related to neuroscience is sort of in the purview of reading some private chat here, and if someone has to tell me something, I'm sure they will. Okay, so I'm just going to keep plodding along here. So NIF's purview is sort of all of neuroscience, and sort of being able to aggregate and identify all the different resources and provide information about those to the user in a coherent and organized fashion. And again, just using NIF and autism as an example, just to dig into that database in a little more specific way. NIF, is responsible for trying to find any data that's related to autism, any literature that's related to your query, any resources, the registry of resources, any funding mechanisms, any web presences that are related to that query as well, and present resources to you that way. And so this is just a quick look at the resources, resource registry return on the query for resources that have something to do with autism. And there's 90 some resources registered with NIF that are related. The Autism Research resource, Autism Speaks, Autism Tissue Program, et cetera, are all resources that are dedicated to or have some link to that particular query. So moving along, in addition to so looking at a different portion of this query, looking at the data that this autism query returns. Again, there's 127,000 records of that have been annotated to have some sort of data representation that have to do with autism data sources. Again, top on this list is the Autism DB, a database of animal models used in autism research. The Internet Brain Volume Database has lots of brain volumes that come from patient populations that met that keyword, et cetera, et cetera. So again, there's NIF tries to identify sources and categorize those sources and make the elements of those sources available that are relevant to your particular type of query. There's also other types of hierarchy that can be done to try to winnow down your results to the appropriate types of things. If you're looking for cell-related information, we can cut down to the data resources related to cells here. If you're looking for function or genes or gross anatomy or molecular information, et cetera, they try to expose and within an ontology, it's the framework to expose these data sources so that the end user can navigate down to the particular type of information they want and identify those resources. So that's NIF, and its mandate is quite broad. So the other sort of contrasting but complementary resource is NITRC, the Neuroimaging Informatics Tools and Resources Clearinghouse. It makes available, information about resources, the specific set of resources that are specifically related to structural and functional and neuroimaging originally, and neuroimaging broadly now to include MEG, EEG, optical imaging, and other sort of neuroscience and clinical neuroinformatics tools that relate to the use of neuroimaging data. So it has a much narrower scope than NIF, and it has but a similar need to first categorize all the different resources that are pertinent to that particular topic. And so this is the front. This slide here shows you the front page of NITRC at nitric.org, and some of the information that's present on that front page is sort of quick jump into the database of resources by domain. It could be MRI or CT or PET or optical tomography, etc. You can browse by different software functionalities, software that does image segmentation or motion correction, etc. You can browse by data resources to find data that's relevant to whatever type of query you do. And you can browse by diagnostic types of characteristics as well. There's other sort of featured tools, and there's general community information. There's sort of recent updates to the content of the site, and there's some news that comes out to again try to keep the community engaged and relevant to your sort of day-to-day neuroimaging life span. So jumping in. This is the result of a query on that site for volumetric analyses. So a bunch of different tools were identified that have claimed to be related to volumetric analysis. And so you can delve into here, find those different resources. You can winnow that search down by saying what specific operating system you want the tools to work on, what particular data types you have, what particular licensing you might want to have. So all those are ways to reduce down all the different resources that come back to you to the ones that are particularly relevant to your own particular needs. Each resource themselves have the sort of standard front page for that resource. This is a program called ITK Snap that does segmentation and image analysis of various sorts. The NITRC homepage for ITK Snap. It's important to note that all this content comes actually from the resource provider themselves. So ITK Snap is providing this information, not some NITRC person describing ITK Snap for you. But in this particular project, you are one click away from the documentation about that resource. You're one click away from downloading that resource. You're one click away from getting to that project's homepage. You're one click away from getting news and other helpful types of things about that. NITRC supports a community ratings and reviews types of options, so that as people find tools they like that install well, that do their jobs well, they can help give them a +1 type of information. You can also see some of the activity that is going on in the resource, the number of downloads, number of publications, and other features of the resource. The nice thing about a NITRC homepage for each project, though, is that they're identical. So while the websites of 20 different software packages will present very similar information to this perhaps, but in a quite idiosyncratic way to the ways that the developers were interested in, the NITRC homepage for each project can point to that same information, but is always in the same place. So you always know where to get to downloads, you always know where to get the documentation, you always know where the ratings and reviews will be. So it homogenizes it and just makes it much more easier for individuals to navigate around the different content. So again, running that similar query that I did in NIF on NITRC to find different resources within NITRC that are related to autism. Again, we find some of these similar types of resources, but again, presented in the NITRC way, in which case the downloads, the ratings and reviews, the pointers to the documentation, pointers to publications, etc., are all made available in that standard framework. So NITRC has a little narrower scope than NIF, and it goes a little deeper into each of the resources, but they work together and share information and sort of make a cohesive pair of resources to do resource discovery. So that's ways to find ways to find software and websites and sort of community forums and hardware and button boxes and assessment batteries, etc. Finding imaging data itself is a little bit different. So where there might be 20 different softwares to do image segmentation, there will be ultimately thousands and thousands of different images that might be relevant to a particular query or that you might be looking for particular imaging data. So the nature of that query is a little bit different. So we need to consider that a little bit more. So one of the second main functionalities that NITRC developed was the NITRC image repository. Now, so again, the idea here is while in a regular project, I could share data with you just by putting a file of data out there, and a tar file, an archive file of 100 subjects, and just make that file there, give you the demographics. You could download that, and good luck at unpacking that and finding the particular subjects that you want. So a more convenient to the end user opportunity is to really do an image database where data are searchable and specific cohorts of subjects can be identified. So NITRC the NITRC image repository is built upon, again, I mentioned the XNAT image archive toolkit from WashU, but in a way that NITRC image repository projects are tightly tied with their NITRC projects themselves. So the NITRC project, the NITRC resource project, can have users and manage their accessibility to the projects. And then there's, when enabled, a link to the image repository that gets you to that particular project's set of data. And the project itself can manage your access. Is it publicly accessible? Is it privately accessible? Do you have to register? And what kind of authentication do people need in order to qualify for access to your particular data project? So NITRC resources that have the image repository enabled have this little image repository link. You go to that and you jump into the XNAT image repository. And in this particular case, 1,000 functional connectomes has some large number of subjects there, a couple, 2,000 or whatever it is. I can't read it on the screen, but you end up being able to access each of those particular subjects. You could click on one of those subjects and you'd see that that particular subject has a particular age and particular gender, has a particular diagnosis, and has some number of data sets that are available. In this case, 2 structural data sets and a resting state data set. And dig further into those data sets, you can see the parameters, the field strengths, the repetition times, the voxel sizes, and other features of those imaging that's available to you. So you can drill down and find different data sets of interest, but more importantly, you can query within specific projects to find subsets of that data. In this case, I did a query within the 1,000 functional connectomes data set for males between 40 and 50 years old, and there's 38 different subjects that are identified. So you can access these subjects. You can see some information about the type of data that's available, and if that's a query that generated data that you want, there's a download link to download that data right to your own desktop for your own use. More importantly, though, is since NITRC itself hosts many image projects, you can actually search across all of those projects. In this case, I did a query across all the NITRC projects for males between 10 and 15 years old, and we identified 980 different subjects that are within that NITRC confines: structural imaging, functional imaging, diffusion imaging. Many different sources of that data. Some come from the Autism Brain Data Exchange, some come from ADHD project, some come from the Candy Share project, etc. So again, this is a way that when the data is under a homogeneous system, queries for particular characteristics can be done. This could again be further widowed down to different field strengths, different working with the resting state data, different common TRs, etc., can be used to widow down just to the particular sets of data that you want. Much more efficient by doing that from a database query than downloading all of that data and then picking through those to find the subset of data that you wanted. Okay, so now what? I found some really cool software from the NITRC resource registry. FreeSurfer made some really, is very popular and does lots of anatomic segmentations. And I found a bunch of data. I found 980 kids there in that last query that I want to do a analysis of kids on the young and sort of the age development of different brain structures across that age group. well, great, I found 980 subjects, and I found a piece of software that takes about 24 hours per subject to run. Do the math, and that's a really long time if you need to do that. So your options, the sort of old-fashioned options, is you could download that software and hope it works on your desktop computer and go through the installation process. You can download that data and hope that you have the space for that 980 subjects to put it on your computers at home or in your own office, and then let that run and hope that you don't have anything else to do for a couple of years it might take to do that particular analysis. Or maybe your lab has high performance computers, has many more cores, or maybe your institution has high performance computing. So you can contact your high performance computing folks and try to make sure that they can install FreeServe and they have the resources for you to do high performance at home. And then the last option, another option, is using the newly available cloud computing resources, as in Amazon and Azure, where you can purchase or lease or rent computing power as necessary. But to do analysis in the cloud, that's another type of opportunity to do these types of processing. So let's look a little bit more at that kind of functionality. So the third leg of the NITRC set of services is the NITRC computational environment. It's available currently on the Amazon Web Services marketplace. So that's a public web page. Just search for NITRC and you'll find the NITRC computational environments that we'll talk about for the next few minutes. These are standard systems that come pre-configured with a bunch of your favorite image processing tools, the FimRib software library, AFNI, FreeSurfer, and many more softwares. It's built on a standard operating system, the NeuroDebian platform, which makes it easy to maintain and easy for tool developers to make tools that are will install easily on it. And it's dynamic in that you can turn them on and off at your will, and it's scalable in that you can use one core or 16 cores or cluster them together into however many cores that you want. The somewhat downside, of course, is you have to pay for that computer use. You're leasing it, you're renting it, so it's a really different cost model. So again, a option, instead of writing in your grants to buy $5,000 worth of computer at home, you can use that to lease computer time as necessary, say in the Amazon type of system. But regardless of that costing, which we'll talk more about later on, let's look at what the configuration of these computational environments look like. So again, from the Amazon Marketplace, searching on NITRC, you find our two offerings, one of which is the plain old NITRC computational environment that runs on a couple of their classes, Amazon's classes of computers. That's here, whoops, wrong way. And then you'll also see our special configurations that are necessary for the compute cluster instances. So when you want to get to more cores, the class of Amazon computers that are available are slightly different. So we had to reconfigure a separate configuration for that. Once you select one of those computational environments, Amazon nicely reminds you sort of what your options are about how to configure it, sort of how much per hour such a system is going to cost, and where you deploy it in the cloud Amazon infrastructure. Get selected, and you give your Amazon authorization, and there you go. Of course, the important thing with computers that you rent or lease is that you really got to be very efficient and good about using them. You're paying for them if they're sitting there doing nothing, or if they're sitting there working hard. And so to make it more cost effective, you want them working hard and then shut off when you're done with them. So again, that's one of the new sort of paradigms of how to do so this cloud-based computing, is how to be efficient in terms of getting the right size computer, getting all of those cores working hard, and getting it shut off as soon as you're done. But once you commission one of these computers, you get a URL, and once you go to that URL, you get the NITRC configuration page for that computer. This is just a web browser onto your own, personally rented computer. You set up some, features such as your username and passwords and some monitoring functionalities. Get set, and then you get to the launch page for that computer, which reminds you all the different softwares that's installed and reminds you the licensing terms of those different softwares that you're agreeing to. And then you can start your VNC sessions if you want to sort of use a remote desktop to the computer, or you can then access it, as well through the remote desktop or just through SSH or other intercomputer communications techniques. So you've selected a computer, run it up, and you can now process on that as you see fit. It comes pre-installed, as I mentioned, with many different softwares that are commonly used, and so it's easy to get the exact same environment, and the installation hassles, if there were any, have already been handled by someone else, the tool developer, as opposed to you figuring out how to install FreeSurfer on this particular configuration. And so just cute little snapshots, again, reminding us of the different tools and resources that are pre-configured on that. Again, it's a NeuroDebian base package. So again, things that we didn't happen to pre-configure on it for you, can be easily added yourself, and you can tell us, and we will package that into future releases of the system as well. So the most important question right away, though, is what is this going to cost me? Because again, I have no idea what Amazon's pricing is. I have no idea what the cost of some of my usual analyses will look like. So performance and good, efficient utilization of these resources is a important consideration. Exactly how quickly you can get a job done depends on how powerful the computer core that you purchase is. Amazon provides a number of different sort of core performance capabilities. How many cores do you purchase? How large a system are you using? How well you utilize it, and how well the software you're using parallelizes across cores, and how much data you want to do. Again, all factor into what it costs to do a particular analysis. And so part of NITRC's job in this case is to help train and teach us all what the actual cost of this will be, so that we can understand better how to compare and contrast the cost of cloud computing versus ground computing at your own local institution. So let's we've worked our way through a couple illustrative examples. I don't want to go into too much of the details of this. This is a particular process that's necessary for diffusion tractographic analysis. It happens to be one of the more time-consuming aspects of it. The bedpost, the Bayesian estimation of diffusion parameters, can take many hours, and is one of the longest steps in a standard diffusion processing stream from the FSL suite of tools. So running it on a particular case that on 60 slices and particular parameters shown here, and taking advantage of the fact that the FSL software automatically parallelizes its software using the SunGrid engine, which is configured on the NITRC computational environments, and will parse those out to the cores as necessary. So it will take your one job, which may take 60 slices worth of probabilistic estimation, and turn that into 60 jobs and split that across your cores. So just looking at the relative performance on my own, one core of my own desktop Mac, this particular bedpost example takes about 5 hours or about 6 hours to process. So that's this little line here. And depending on the exact configuration of the Amazon compute environment that you use, you can get something that's slower than your Mac desktop and cost you $2 to run, or you can get something that's faster than your Mac desktop that can be done in about 20 minutes and cost you about $3 to run. So this is just one subject's worth of analysis, but you get it done in 20 minutes. You're paying by the hour, so it's actually 3 subjects you could have gotten done in that. So that runs out to about $1 per subject, and you can do 3 subjects an hour. If you use this particular 16-core system. So that's just one example. And so again, if you have hundreds of subjects, you can begin to calculate what the cost will be and what the time will be. So again, going back to my little example from before that said I wanted to do about 1,000 cases, and it's about $1 a case, well, then it's going to cost me $1,000. Now again, that's on one hand seems large. It's $1,000. On the other hand, if one thinks about the alternatives to doing that, and purchasing that through your grants and things like that, as opposed to purchasing computers or funding the depreciation of your own institution's high performance computing, again, there's still questions about exactly how to best use such compute dollars. But the other good thing about this same $1,000 is I could do that, if I just use that 16-core system I showed you on the other page, that takes about 20 minutes per subject. And to do 1,000 subjects, that's going to take me 14 days. And so 14 days is better than the year it would have taken, or 31 days that would have taken on my own desktop. But for the same $1,000, instead of using one 16-core computer, I can use the star cluster, the elasticity of the Amazon system, and cluster together 333 of these same core components and generate 5,000 cores and get this whole job done in 1 hour. So again, for that cost, one has a lot of flexibility about how quick and one can do that depending on how the job scales and parallelizes. All of this pricing I've done here is on their on-demand pricing, which is the higher pricing tier. With other trickier thinking, one can also do spot pricing with Amazon and get significantly lower costs at the cost of some uncertainty about exactly when they will run and when you get computers at that cost. So these are sort of upper bounds of the cost of some of these things. So again, a bedpost costs about $1 to do. Similar experiments on a voxel-based morphometry of 100 subjects will cost you about $3 to do, and about 8 hours to accomplish that on a particular configuration with 8 cores. FreeSurfer, which can be done on a small 1-core system in 20 hours, will cost you $2 or so. And again, you can make that happen faster, but again, that's kind of the order of magnitude of the processing that's available for that kind of thing. So at least that begins to develop situations to figure out sort of what the costing of these different opportunities will be. So last but not least, I want to get into a couple sort of real-world use cases of this, in the application to both doing sort of more comprehensive big data elements of publications, and also some processing that's begun to be done with the computational environments on the NDAR database. Without getting into too much discussion, a lot of our current processing and thinking, goes towards the journal article being the most important thing. That's what gets you your grants, gets what gets you your tenure, gets your improvement in life, and the publication is really based on some interpretation of some results which are handpicked to tell your story, which came from one particular analysis method of many that one could have done on a big huge pile of data that one has. So the only thing that persists currently is that interpretation. Your journal article says the caudate nucleus is big and correlates with the behavioral dysfunction in kids with behavioral dysregulation. And that's what ends up in the literature. That's what people cite, and that's sort of all that lasted from a huge pile of data from one particular analysis technique that you took versus one particular set of results that happened to turn into the story that you chose to write about. A lot of this big data effort is to try to turn that upside down. Yes, you still get your journal article down here at the end, but it's that data which is the most long-lasting part of it, especially when that's shared. Yes, you can do your particular analysis, but other people could do different types of analyses. Making your results available, yes, you have to pick some results to write the story, but we need to make it so that all those results that came out are available as well, so that when you come to your interpretation and your journal article, we don't lose this whole information that led into that. And that's amongst the things that sort of drives the big side of big data and big computation in cognitive and behavioral neurosciences in general. So right now, again, it's not rocket science to do any of this. A lot of the technologies for integrating data into your publications exists now. Looking here, not to read the details, but a small version of a PubMed entry from a particular paper of ours. PubMed has these link out options you can pick on, and the user using different techniques can make these link outs appear for your own particular publications. So this particular publication has a link out to a NITRC project page which hosts all that data that went into that. So the raw data is linkable to the publication through that link out there, and then also the the meta-analysis. In this particular paper was volumetric analysis of a bunch of different structures in a particular patient population, and that can be linked directly to the publication as well. So again, in this particular situation, my publication and PubMed and my conclusions are here, but also your linkage to the raw data and your linkage to the metadata that's associated with that can be established. So again, as long as one's willing to share their data and put them someplace that's findable, the linkages, the connections between your publications and your data and metadata can be established. This is gory details of not only the data that was part of that publication is available on this particular website, but lots and lots of other things about that data can be made that goes even beyond the analysis of that paper, and other subsequent analyses of that data can also all be linked together. So that's a good thing to do. So to finish quickly, again, looking into the NDAR database, the National Database for Autism Research at ndar.nih.gov. This is part of the National Institutes of Mental Health's effort to really consolidate their research prospects in autism under sort of a unified situation. The database itself hosts all sorts of information about phenotypic descriptors, about genetics, about neuroimaging. We can dredge down into this and again find all of our 6 to 12-year-old boys yields again some set of sessions with some set of diagnostic characteristics and some set of behavioral characteristics. We can dig down into that and request that data once you have proper authorization. And so not only did I find that data based on its imaging, the fact that it has imaging, there's hundreds of other measures and hundreds and tens of different sites where that data came from. That is all available to you once you did that query and do that download. Once you download that, you can get a data package that again you can run at home as before, or run on any high performance system or on the cloud. That data lives also in the Amazon S3 simple storage environment, so that instead of downloading it all, a little relational database can give you a pointer to where that data resides, and so you don't have to even download things when you're working in the cloud environment. And then different pipeline and automated systems can also be invoked on that. So yes, you can handle that by hand, but you can also use something like the Lani pipeline, which is a workflow environment manager to set up workflows that take the cloud-based source, the S3 source of your NDAR data package, and then do different processing. In this case, a free surfer processing and an FSL processing, and you can launch those to run on your NITRC computational environments or on Lani's high performance computing. And so again, this is a way to access the NDAR data, to use your NITRC computational environment, and to use a nice graphical interface to interoperate with that environment to run and control a large analysis. So that's been done on the large pile of data that's in the NDAR data set. 2 different types of runs have been done, one of which is a image quality assurance checks. A bunch of different signal to noise, contrast to noise, volumetrics types of measurements have been made across that structural data. There's a diffusion quality assurance and a functional quality assurance that's done as well. So these have been run using the NITRC computational environment. These results have been fed back into the NDAR system, so those then become queryable as parts of the features of data that you select based on structural properties. And not only has the quality assurance been done, again, a common thing one might do is run free surfer or different types of structural analyses on this data. So again, as those get run centrally, if one wants, that data gets put back into NDAR, and that becomes properties that you can navigate against the data. So that's really where I think I'm going to come to an end in general. In summary, big cognitive neuroscience these days is evolving to include data repositories and management of large data, management of workflows and analysis systems in a reproducible fashion, management of the results into metadata stores so that all the results can be retrieved and queried against, and not just what shows up in the words of the literature. And then of course the interpretation and story written about the data will continue as it has. So again, each of these types of information require tools and resources to get your data into it and to interrogate the data that is there. And so we all have to go through these new learning curves, to be able to wield these things efficiently. I'd like to thank, the NITRC enterprise for helping, make this happen, and a number of, co-investigators I have both at UMass, at Turner Consulting Group, in Washington that does the core NITRC development, the NDAR folks, and the Lonnie folks for helping us use the pipeline in this environment. So that leaves me a couple minutes left for questions. I think I'll leave it on this slide for the end, and I'll see if I understand how to see if any of you out there have questions. I see no questions yet in my question box. It was either, oh, great presentation. Thank you. Do I see that? So I guess that was supposed to be a question mark, not a exclamation point. But so was it a great presentation? No. So again, try to get a bunch of information out there for you, and again, resources to be useful. And again, contact me or any of the other resource providers to help you. Why doesn't the NIH mandate all researchers to share their data? Well, they're beginning to, and again, I think they've been starting, slowly. So from 2008 or so, the initial data sharing mandate came out that said all grants that are larger than $500,000 annual direct cost budget, have to have a data sharing plan. So that was a good start, but that left a lot of openings in it. So most grants are smaller than $500,000, so that kind of evaded that. And having a data sharing plan doesn't necessarily mean it's a good plan, or that the data is well shared. And most importantly, there was very little teeth in that mandate, in that you could write your plan and it wasn't a scorable element, and no one checked your data sharing very well afterwards to see if you complied. So I think the NIH is working on beefing up those mandates, and there's data sharing groups within the NIH who are working on that. And also initiatives like NDAR. So within a NIMH who can exert some control on their autism portfolio, they actually do. It's part of their contracting. It's part of the Autism Centers of Excellence. It's probably written into virtually every autism grant that says thou shalt share all of your data, like it or not. So again, I think slowly but surely the mandate to share is coming along. The counter to the mandate to share, though, is also having resources to share your data in. So things like NITRC, things like these XNet databases, they cost money. They're infrastructures, and every dollar NIH spends on infrastructure is a dollar that's not getting spent on solving autism, or solving bipolar disorder, or solving other things. So it's a it's a tricky question that again, mandate to share, but making sure that there's places to share is also important. So again, these are tricky philosophical and things that are being worked out and are improving, and again, more resources are coming available, and more ways to figure out how to fund that, and sort of the data sharing tax, make sure that's not too high, and make sure the value for that is worth it before you force everyone to do it, or else the peasants will revolt. Okay, just 2 minutes left, I guess. And I thank everyone for attending. It was fun to try this electronic presentation, and I hope that was interesting. Share that data.